Hi everyone, welcome to this CSIR Life Science important unit and topics discussion session. So here I am listing out some important topics and which units are very important, which units are having some least important. Based on that, we are segregated total 30 unit is there in the syllabus. That syllabus I divided into three category: most important, important and considerable. Okay, so most important, that is fundamental processing, that is third unit of CSIR. And next it is system physiology of plant, and next animal physiology. Okay, so then inheritance biology and methods in biology. These 5 units are most important whenever you are considering for preparation. Now we have a short period, so in this short period, if you are focusing on this 5 units, then we will get 50% of the score, because it's covering all the these units. It's easy to study, and as well as you are expecting more questions from this unit, easily you can answerable. And next set of unit is the it is important topics. These units are bigger units, but few questions only they will ask. So this first one is the molecules means everything will come: atoms, DNA, RNA, protein, everything. Okay, that structural interactions we want to study. It is larger one, and it is a unit 1. So second, cell communication and cell signaling. This is also we have many pathways. And fifth unit, that is developmental biology, so that is, and then ecology. So these two, okay, unit 5 and 10, it is having some importance, but less. If you are referring some topics, you can easily answer and get some marks. Okay, so this first 5 and next 4, it is having most important values whenever you are studying at last minute. Okay, so if you are starting, if you are started already, means not a issue. Now you are freshly starting for preparation, so within this 1 month how you can clear means, then first focus on this first 5 unit: unit 3, unit 6, unit 7, unit 8, unit 13. And then in this also I have some topics, because we can't able to cover whole topic, right? So if you are focusing on few important type topics, then easily you can score more mark. Next, you consider this 4 units. This 4 unit 1, unit 4, unit 5, unit 10. First focus on developmental and ecology, because that is new to us, normal biotechnology people and life science people. So this is, you can cover some topic and get more mark to attend that. Easily you can attend more questions. And then focus on the cell communication and molecular inheritance, molecules and then interactions and all, you can go. And next 4 unit is the it is wide topic, but it is having the you can't predict which topic is important, so this you can't able to cover the full topics. So take unit 2, unit 9, unit 12, unit 11, that cellular organization, diversity, and then applied biology and evolutionary theories and behaviors. Okay, so these 4 units are considerable. And this is also because every 13 units you should study, all 13 you should study, but you just categorize based on the priority. So give priority to the first 5, next priority to the fourth, for this 4, and then next last least priority to the last this 4 units: cell organizations, other that's organs and all, and diversity, applied biology, and evolutionary behaviors. Okay, here I am listing out some topics which we want to focus. Okay, so here I am listing out that topics. So in that unit 3, fundamental process, okay, that is the first topic, most important topic in the our syllabus. So okay, that is RNA replication, transcription, translation. This central dogma, you should know the enzymes and its products. Okay, that based on that you can expect. Then so this is the these 2 steps are major important in the DNA synthesis and protein synthesis. Then DNA type, RNA type, DNA damage, repair, mutation systems. So RNA processing, that is post-transcriptional modification, post-translational modification, inhibitors and topoisomerase inhibitors. And one OPRON model is very important. And gene regulations, what are the gene regulation in the prokaryotic, eukaryotic and virus, phage, everything we should study. And chromosome, chromatin, it comes in the gene expressions, okay, gene expressions, and genome organization, it comes in the chromosome, chromatin. And energy conservations and mode of action of antibodies, and experiments, which is this Messelson experiment, homologous site specific recombination, and some enzymes, this polymerase, role of the RNA polymerase, role of DNA polymerase, kill now enzymes, and histones activity. These are all the important. Next, plant physiology. In this plant physiology, some important topic is there, because it is very short and easily you can score more mark. Okay, so first one is the water potential concept and how the xylem phloem are acting. And next, photosynthesis is a dark condition and light condition. This C2, C3, C4, CAM pathway, glycolysis, TZ and all, it's very important. All pathway you should study. And electron transport chain, phytochromes, photochromes, cryptochromes, that everything is important. And nitrogen ammonia assimilation and amino acid synthesis in a plant, and biological system for nitrogen fixation. This phytochemical, this is very another important topic. From this phytochemical they are asking more questions. And stress physiology, biological clocks, stomata opening closing. This stomata opening closing also important. Okay. And ABC model, another ABC model, definitely they will ask one question from this ABC model of flowering. And next portion important is the system physiology of animals. Here it's consider all the system. First two is considering the blood circulation system. So blood, how it is in our body, how it is created, how it is involving in the function. And then next we should know the cardiac system. Okay, so this cardiac system, and then endocrine system, excretory system, so hearing and vision system, reproductive system, and that excretion system, waste elimination, and it's these two is comes in the excretion. And apart from this we have vitamins deficiency and disorders, hormone deficiency and diseases, and neurotransmitters activity. This and all we want to study. So, and after this 3 is totally different. For it is from this definitely they will ask one question. And transport of gas in the lungs, how it is transporting in the lungs, and digestion and absorption system. These two systems are also very important. Okay, so 15th one is lungs, 16th one is the digestive system. So these are the most expected part in the our system. Okay, animal system. And next topic is the inheritance biology. In this very few topics they will ask more questions and we can get more score also. So pedigree analysis and gene disorder finding, and monohybrid, dihybrid crosses, population genetics, chromosome alterations, mutations, linkage markers, physical mapping, and complementation and gene dissection. Molecular markers is very important topic again. Molecular markers very important. And inheritance and maternal inheritance types, and co-transformation experiments, tetrad analysis, and QTL mapping. This is based on that they will ask the part C question. Mendelian experiments, topoisomerase inheritance, and fragile X chromosomes and gametes related questions they are asking in the analytical questions, part C. Next part is the methods in biology. Is very important. It is a vast one also. So here methods in biology. So first we should know what are all the molecular biology techniques mainly they used. Okay, so that cloning, vector, restriction digestion, CRISPR technique, restriction digestion, enzymes, what are they using, recombinant DNA technology, blue-white screening method, how they are after the cloning, how they are screening, electrophoresis, all 1 dimension, 2 dimension, and STS page, isoelectric focusing method. Then DNA sequencing, Sanger sequencing, Maxim Gilbert, whole genome sequencing, metagenome sequencing. The differentiation we should know. Then parametric and non-parametric test. What are the tests we are having in the biology? And what is the use of plasmid? What is the use of plasmid, DNA, bacterial artificial chromosome, yeast artificial chromosomes, and plotting techniques, fish gist, DNA proofreading methods, and PCR. Definitely they will ask one question from the PCR. Okay, PCR is very most important topic. And ELISA, another method. ELISA and FACS also very important. Next, chromatography techniques and certifications, UV and NMR X-ray, that methods they will ask. Next, electron microscopy, it is also very important. SEM-TEM, from this they will ask one question. And gene knockout method, phage display, nick translation, patch clamp method, yeast to hybridization method. These are all the previous year they asked questions from this topics. That's why here we are listing out. And this physiology, how the biomedical methods we are applying, we are using in our analysis. These are all the biomedical methods. And mutagenesis deduction, in tissue hybridization method, and here the chromatography and certification is another type. This is diagnosis also important. Okay, what are the kits they are using for the kit method they are using for the diagnosis. And next, some important topic. This until this most important. Hereafter some important topics. Okay, so unit 1, the molecules. This pH concept is very important, and Henderson equations, amino acids, PI calculation method, Ramachandran plot is most important. Definitely one question from this it will come. And then electron transport chain inhibitors, it's most important. Protein analysis in system, enzymatic cleavage by the protein, which enzymes are cleaving the proteins. And then from this enzyme, okay, enzyme concept, you can expect 3 or 4 questions from the enzyme concept. Okay, and then protein folding, degradation, cofactors, DNA structure. This kind of things we studied in the unit 1 also. So unit 3 also, that is recalling that process. And DNA conformation and nucleic acid synthesis pathways. Okay, de novo pathway we should study. And Gibbs free energy related calculations, thermodynamics in the biology system. This cycles, okay, this all it is see, Kelvin cycle, Krebs cycle, TCA, fatty acid oxidation, HMP pathway, Kelvin cycle. These are most important. From this you can expect one question. Okay, next unit 4, cell communication and cell signaling. Here I am listing out, that is a big topic. From this I listed out few topics only. So in the cell communication, host pathogen interaction, how it is interacting, and entry of virus and pathogen induced diseases. This is last 3 years they are asking continuously from this. And then bacterial evasion mechanism, cell mediated humoral response, antigen processing, complement system, emerging concept, innate and adaptive. These are all general, just recall it before you are going for exam. And autoimmune diseases, SARS-CoV related updates, and cytokines, vaccines. From the vaccines they will ask one question. And antibody antigen interaction, immune tolerance, cancer. Okay, this cancer concept is another important. Okay, definitely one question from the cancer cell cycle or cell adhesion or molecules. Okay, from this they will ask one. Next, cell maturation, cell junction, cell interaction, apoptosis, oncogenes. This 4 are this 3, okay, specifically these 3 topics are very important. Okay, these 3, apoptosis, type of proteins, and oncogenes, tumor suppressor genes, this pathways, JAK pathway, RAS pathway, RKT pathway, GPCR. From this they will ask one question. And rhodopsin and delta notch pathway, and nuclear receptors, map, monoclonal antibodies, polyclonal antibodies, hybridoma technology. Hybridoma technology is very important. Just basic questions you just refer. Next is a developmental biology concept. So here Drosophila development, this first 5, okay, Drosophila development, vulva formation, and fertilization of sea urchins, limb formation, cleavage concept, gastrulation. First 6 concept is very important, most important, and apart from that developmental biology, some important concept is there. That is the organogenesis, paracrine factors, frog development, C. elegans development, C. urchin development. This 3, okay, from this 3 this 3 theory is very important. Okay, next RAM SAM activities, and then plant developmental ABC model already we studied in the previous itself. So ABC model is very important. Okay, then neuralization and sex determinations, trophoblast and stem cell types which they will ask one question definitely. And next one, it is ecological principle. It is very big topic and important. So it is niche and habit, first 5. Okay, this here also the first 5 is very important. From this niche, population ecology, top down method, bottom up, and species interaction, community ecology. From this we can expect 2 or 3 questions. Okay, next is the succession theory is the most important one. Island biography theory also important, and conservation biology, where they are conserving, and our Indian conservations, what are the Indian conservations we should study, and energy flow nutrition cycles also very important. This here I am listing out, not listing the whole syllabus. I am listing what are all the important topics alone. Okay, next one is a considerable topics, that is cell organization. So in this, if you have a time, study the whole syllabus. Otherwise you just check this few topics alone. The chromatin structure, cell organs, transport, how it is happening across the membrane, cytoskeleton structure, cell cycle, checkpoints, protein folding and sorting, lipid draft, what is a lipid draft, and transposons, P53, PAM3 pathway, FRAP pathway, and activities, plasma membrane, how it is acting, osmosis, ion channels and pumps. From this they will ask one question, osmosis, ion channels, pumps. Okay, and next endocytosis, phagocytosis, difference, virals and bacteria growth, how it is growing in the nutrition, what are the nutrients which required, and diversity, it is a big one. In this first you focus on few topics, that is the taxa of the plant and classification, and phylogram, cladogram, image based question they will ask. Then conservation strategy, endangered and rare species, they will ask definitely one question, and parasitic pathogen interaction. Next, adaptive modification, how the animals are adapting and modifying the system, and apart from this, habits, types, and iconic species, who are all the, which animals are iconic species, and everything we should study. And crop pathogen, it is this is the important topic in the exam aspect. Okay, next, strategy and policies in a ecological system, what we are following. And next topic is the evolutionary and behavior. It is a final topic which we are focusing on this. Okay, after that we have applied biology. Here this is a new one, evolutionary and behavior. So here natural selection, speciation is very important, and then Hardy-Weinberg equation also very important. Kin selections, it's a most important topic, and nuclear theory, Mendelian integration, Miller experiment, metabolic evolutions, and then molecular clock theory. Okay, clock theory, sequence analysis, adaptive radiations you should study, and this co-unit, evolution and behavior unit. So first part is covering evolution, second part is covering the our learning, memory, and cognition, sleep. This mechanism comes in the behavior. and then social communication, how it is acting, and mating system in this ecology, and optimal habit selections, migrations, how it is happening. These are all the important topics in the unit 11, behavior and ecology. Next is the final one, applied biology. It is having more topics. Okay, it is you have to cover more content and less questions only they will ask. Marker assisted selection is the most important. As I said, molecular markers, gene transformation method, agrobacterial mediated gene transfer method. Definitely they will ask one question from this agrobacter. Okay, next TI plasmid and repeated sequence, that's roles, and protoplast fusion, how it is happening, and transgenic plants related, they will ask one question definitely. Okay, next breeding in animal and animal cell culture related questions they will ask, and then microbial metabolism, vaccine productions, bioremediation, and industrial waste and bioleaching. Last 2-3 years they are asking questions from this. Okay, next bioremediation, phytoremediation, biosensors, seafoods microbiology, and gas biosurfactants. So this is another important portion in the unit 12. Okay, applied I think 13, applied biology. Okay, so here I am listing important topics, first important units, that what are all the topics are most important. In this whole syllabus I am not mentioning the whole syllabus, not included the whole topic, whole syllabus. This is for last minute's preparation. Which topics is most important and high priority. Okay, they are asking more questions from this topics, easily you can answer, get more mark, and that's why we are listing this units. First cover first 5 units. Okay, so fundamental process, system physiology, system of plant, animal, and then inheritance biology and methods. Okay, then go for the remaining units. Okay, if we are covering this, definitely we can get the more mark and we can clear the CSIR examination. So you just refer based on this priority, then get more mark. All the best. Thank you.